Okay, thanks. Okay, it's great to be here with all of you today, and I don't entirely know what you've learned up to this point, but I'm going to go over some methods, probabilistic models for population structure, and talk about some models that my group has developed, but mostly try and focus on the differences in assumptions that each of these models make and what they actually look like when applied to genomic data. So please, this is supposed to be very interactive, so stop me anytime. And to those of you who have seen any of these slides before, I desperately apologize. Okay, so one thing that I think is particularly important that I'm sort of excited about thinking about recently is exploratory data analysis. So a lot of statistics focus very much on hypothesis testing, and I think that's a real shame. I think a lot of the things that we do in genomics is exploratory, where we really would like to find trends, patterns. We would like to develop the hypotheses given the data that we have. Often, these exploratory data analyses are visual in the sense that we can take a matrix of values and, for example, plot the first two principal components and see how each of the data points cluster. Okay, and then the idea is that looking at the clustering, we can start seeing things that pop out here, like, for example, that if you squint, looks quite a lot like a map of Europe. Okay, contrasted with hypothesis testing, which is sort of the bread and butter of statistics for the last 150 years. So in hypothesis testing, we basically have, again, this matrix, but then we have a specific hypothesis here. We have a specific question we'd like to test. We put it through some hypothesis test. We get some sort of test statistic, and we can compute a p-value based on that, assuming specific types of calibration. And this is a really nice and straightforward thing to do. No one really, I mean, besides p-value hacking and all this sort of thing, it's not super controversial. You can basically design a nice experiment, collect data based on a specific hypothesis. You define the null and alternative hypotheses clearly, and then you can test the statistical significance for the specific data of that hypothesis. And then if you have support for that hypothesis, in particular if you have the ability to reject the null hypothesis, if p is lower than some threshold, then you can try to convince people that your hypothesis is actually indeed probably going to be true. I would argue that exploratory data analysis is undervalued and underappreciated in science and statistics generally. And the reason I think it's undervalued is because it's incredibly difficult to do correctly. Okay, and hopefully today when we think about how to analyze genomic data, we will talk about some ways, some caveats, but also some ways to use it in sort of a responsible way. So many assumptions in exploratory data analysis are not well characterized, and you'll see today that for every single method we talk about, I'm definitely going to continue to hammer home all of the assumptions that method makes, because it's absolutely essential to understand what assumptions are going into the patterns that you're eliciting from the data in order to be able to interpret those patterns appropriately. In general, it's not the same as hypothesis testing in that there's no null hypothesis. Okay, there's no way to really describe what the data look like without the pattern underlying the data. Okay, if there were, then we could start thinking about p-values or likelihood ratio tests or Bayes factors. But since there's no obvious null hypothesis, this is a very, very difficult thing to do. There's no test of statistical significance. So whether we're talking about PCA or factor analysis or anything else, there's no real way to describe how statistically significant the patterns that you're finding are. There are a few ways to think about it. So PCA, for example, has a way to consider p-values for each of the principal components pulled out. But there are major problems with these methods, and in my opinion, and it's very hard to compare specific patterns found in the data across different samples and hypotheses. Often the conclusion is based on qualitative evidence. So again, you're going to look at a picture, you're going to look at a visualization, and come up with some conclusion about the data, as opposed to actually having a p-value associated with that. So for example, when you look at the if you guys know about this TNAS paper from a couple years ago from Mike Snyder's group, where they clustered mouse and human tissues, and using a visual interpretation of a 3D PCA plot, they said, well, it's pretty clear from these data that the mouse tissues all cluster together and the human tissues all cluster together. Of course, there were batch effects that they didn't control for, but again, it was a mostly visual conclusion, which is a difficult thing to swallow if you're a statistician, I think. The results are often really hard to validate and reproduce. So this is absolutely true, and there's a number of reasons that this is true. Number one, because of the methods themselves, but also because of, specifically when I say the methods themselves, I mean the invariances in the methods. So we're going to see today that a lot of the methods that we're talking about, whether it's PCA or factor analysis or latent Dirichlet analysis, come with a bunch of sort of rotational caveats that we have to be very, very careful about how to understand and interpret. But also, they're difficult to reproduce because of technical effects and batch effects that may really affect the patterns that you see underlying the data. And if you consider multiple data sets that have different types of batch effects, different types of technical effects, then you could have a really hard time reproducing the signal if it was partially correlated with those effects. Any questions? Any thoughts? Okay, so here are some examples of exploratory data analysis, and we're going to be focusing today on admixture models and population structure, but I just wanted to show you some of these ideas. So you can think about different ways to explore data. So people have used heat maps a lot, right? So this is actually from Gilad and Mizrahiman in 2015. These are two pictures from that. And here you can see a heat map where basically you do this clustering and you have a dendrogram then describing the relationship among all the samples in there. And this is actually the counterpoint, the response to the PNAS paper with the mouse and human tissues where they actually controlled for batch effects. And then you can see along the different axes now that tissues and not animals, species, cluster very nicely after you control for batch effects. And actually the point about, again, this exploratory analysis, the conclusion is, again, visual, where they say, actually, when we control for batch effects, you find an entirely different grouping than the one that they found when you don't control for batch effects. And their point was that because it's so sensitive to whether you control for batch or not, we can't actually make any conclusions. So be very, very careful about what you read from these analyses. I think that's a good conclusion. Principal components analysis, again, is another one where you can look on the PC axes. You can plot all of the data points on the different axes, and you can see nice clustering. And again, here you see that in general, again, eyeballing it, that mostly the tissues are going to cluster together as opposed to the different species in terms of thinking about where each of those points are next to. So visual conclusion. So we're going to be looking a little bit at PCA plots like that. Another type of exploratory analysis that we're going to be thinking about today is admixture models. So just to orient you to one of these, they're called destruct plots for one reason or another. Along the columns are all of the individuals, okay? The rows are, each column, you can imagine, goes between 0 and 1. And each color represents one latent component. So these are called admixture models because the idea is that each individual, which is represented again by one vertical bar here, is going to be composed of some sets of these clusters, right? So some people are mostly one type, one latent factor, one component, and some are a broad mixture across the different components. And you can see that by looking at the clusters of these individuals, the clusters of these individuals across these different colors of these destruct plots. So, for example, here, again, you can see this is the HAP map data, HAP map phase 3. And you can see that in general, with a few population exceptions, a lot of the individuals in this study are not admixed. In other words, they're mostly from population, right? According to this particular representation of the data, where k equals 6. In this PopRes data, this is 1,000 European samples. You can see that basically everyone looks admixed, right? There's no individuals here that are only one of the colors, only one of the ancestral populations. Okay, so that's how you might interpret these plots. Questions? Okay, so we're going to be talking about factor analysis as a general framework for exploratory data analysis, and thinking about this in the context of genomic studies. So we're going to be thinking about, and I should say I have way too many slides, as Eleazar knows I always do that. So at some point we're just going to stop talking and conclude. But Just stop me when you're done. Okay, so we're going to think first about studying latent population structure, which I think is an absolutely critical reason to do exploratory data analysis, and probably the reason that I got into these ideas to begin with. Next, we can think about understanding the genetic basis of high-dimensional traits, and the trait that we can consider is RNA sequencing data or gene expression data. If we have time, we can think about differential and context-specific interactions, which is just a slight variant of the factor analysis model that we're going to be using for the first two. Okay. Okay. So let's basically think about everything that we're going to be talking about, all the models, all the methods in terms of Bayesian factor analysis. And I understand that for some of the methods like PCA, it's kind of a stretch, but let's just put it in this framework for now and keep going within this framework just so that we're all sort of on the same page. So the idea behind Bayesian latent factor model is that we have Y, which in this case is going to be a bunch of individuals on the rows and a bunch of SNPs on the columns, okay? And the idea is that we're going to decompose the individuals and the SNPs into these much lower dimensional matrices, lambda, which is going to be our loadings matrix, and X, which is going to be our factor matrix, okay? Then we're going to have some Gaussian noise, epsilon, which is independent and isotropic noise for every single one of those elements Y, okay? So the idea then, if you think about any specific element of Y, right, Yij, so it's a single point in Y, it's going to be distributed as a Gaussian over the sum of our K latent components in lambda and X, okay, of these scaled, so we're going to scale all of our factors by lambda, and then we're going to have variance, or precision in this case, equal to this phi variable, which is going to describe the residual noise, okay? Any questions about that? Yeah, you seem like you have a question. Yeah, yeah. Is this okay? Yeah.
You're dragging something on the floor.
Oh, I'm dragging something on the floor. Oh, that's my microphone. Thank you. Wait, is it? Oh, that's really odd. The microphone fell off. Okay, I don't know what to do about my microphone. Yeah, that's a great question. We're going to talk about that. Yep. There are a couple answers to that, and I think Eliezer disagrees with me on some of what I'm going to say, but there are models that deal with the categoricalness of SNP variables. There's one from John Story's lab recently, but in general, actually, we find that the methods that we use with a Gaussian assumption actually do quite a nice job because they're often over-dispersed, and so it's nice to have an explicit representation of the variance term. We can argue that, though. I'm happy to argue it. Okay. Any other questions? Okay. So some of the examples of latent factor models that we're going to be talking about today include principal components analysis. So the idea, we're going to go through this very slowly, but the idea is that principal components analysis can fit exactly into this model where they have orthogonal factors and orthogonal loadings as well. And I should also say that for PCA, the covariance terms are going to be slightly different as well. So we'll talk about that. For admixture models or latent Dirichlet allocation, we have the factors and the loadings both sitting on a simplex. The simplex is numbers that are non-negative and sum to 1. So both the factors and the loadings individually are all going to be non-negative and sum to 1. Okay? That's nice in terms of interpretability. Again, we're going to talk about that later. Non-negative matrix factorization, or NMF, deals explicitly with count data. So this is something that would be very nice for the data that we see in terms of SNPs, which are 0, 1, 2s generally. So that ends up working very well, although to be honest, I tried it many, many times with our data sets and didn't find much of an improvement over the other methods that we have, specifically LDA. And in fact, there's actually a very nice tight relationship between LDA models and non-negative matrix factorization. Factor regression models are another thing that are kind of nice. So thinking about it in this probabilistic framework, it's very easy to expand this model to add fixed effects, known covariates, to this model to be able to control for specific things in the data. So if we know that there's going to be batch effects in Y or something like that, or we know that there's going to be sex effects or something else that we want to control for, we're just going to put that in with a fixed effect term. Okay? You guys are quiet today. Okay, so there's obviously going to be a lot of difficulties, as I said, with latent factor models. Okay, and this is where the caveats start to come in. So the reason we should be very, very careful about interpreting the results from these models is because the likelihood of the data in these factor analysis models is going to be invariant to a number of different things, including orthogonal rotation, scaling, and label switching. Okay, so in other words, if I take this matrix lambda and I rotate that by orthogonal matrix R, I can do the exact same thing with the inverse of R to X, right? And all of a sudden, I have two rotated matrices that have exactly the same likelihood. Okay, so that should start making you a little bit nervous. Scale, and actually, I should say, taking a step back, I should say that we're going to see this coming up in one of the examples that we see, where we have a lambda and an x. Every time we run latent factor analysis, we actually get some rotation of the components, and you can see it very, very clearly. Scale is another one, where you can essentially scale each element of lambda and scale x by the corresponding inverse of that. And all of a sudden, you can no longer compare the scales of lambda or x in absolute value, okay, or across lambda, okay. So I should say too that for each of these things, there are some types of priors that you can add that will make the posterior invariant, although it's not foolproof. Let me see what else. Label switching is another one too, where every time I run this, and this is sort of a boring one, but every single time I run this, my k factors here, my k loadings in this case, can be swapped, right? Corresponding swaps on x. So there's nothing that specifically orders them in the models that we're talking about, except for PCA, where it is specifically ordered by proportion of variance explained. But in these general factor models, there's no explicit ordering, okay? So these invariances have a lot to do with, or affect how you are able to interpret these models applied to data. So in general, when we're not talking about PCA, the factors will not be orthogonal. In fact, they may be very well correlated. Okay? So this sounds like it's a problem, and it is a problem for specific applications. In particular, it's a problem when we think about correcting for latent structure in association studies, for example, using latent structure estimated from these models. And that's a problem because if they're not orthogonal, then they could be double explaining some types of variance, and you could be double controlling for that type of variance. The factors are not going to be comparable within or across applications. So if you think about plotting these factors where you have the samples on the x-axis and the scale of that factor on the y-axis, the scale of the y-axis is going to be completely meaningless. Okay? No approach is going to quantify how likely a factor is versus the null. So in other words, how to actually choose k for these models, and how do you know when a specific factor is sort of meaningful, when that type of factor is representing some true signal in the data as opposed to maybe batch effects. And actually, a similar point to make here is that each of these factors that we come up with are not representing a unique contribution to variance. So when you think about PCs, for example, you might think that one PC might correspond to something very specific, right? It might correspond to, for example, the platform that your DNA was assayed on, which is actually often the first principal component if you have two different platforms. It's not ever going to be uniquely that. The problem is that there's a lot of correlation in different types of contributors to variance, and it's never unique. So it's impossible to label these factors with a specific sort of reason, if that makes any sense. Okay. And then finally, I think my main caveat for these exploratory data analysis methods is that it's really hard to be able to interpret any sort of downstream analysis with the factors that you come up with. So visual interpretation is great, but outside of data exploration, it makes me very nervous. So for example, we see people who have, a Y matrix might be gene expression data or let's say gene expression data. And the factors may correspond to particular, again, types of variation in that gene expression data. And the problem is that often those latent factors are used downstream in association analyses. So in other words, they'll take the X vector, which now represents for maybe a set of genes in lambda, maybe it represents something that the researchers are calling the contribution due to effect of this drug, right? This is a set of genes that change when this drug is applied to these data, okay? So they'll take X and they'll use that as the phenotype, okay? But again, for all these invariances, for the reasons that we've just discussed, using that phenotype is incredibly difficult to do because it's very hard to reproduce across studies and because it's unclear that that label is unique and because of the rotational invariances, obviously, too. Okay? Questions? This is very downer talk. Okay. So how can we build a better model? So we would like to actually be able to use exploratory data analysis more robustly on the genomic data that we have. So if we could design the sort of ideal factor analysis model, what would we need? So number one, we'd need it to be robust to sample bias. And the reason I'm mentioning this is because principal components analysis is not robust to sample bias. If I have triple the amount of, let's say, African individuals in my study, in a map study, for example, then a much larger proportion of the variance explained will be taken up in the principal components representing the African individuals. I'll show you examples of this later on. Rotational non-invariance is another problem. Again, we'd like to recover the same rotation every single time. So when we run it 1,000 times, we always get the same rotation because rotational invariance makes everyone nervous. Non-orthogonal factors. Actually, PCA, again, we're going to have orthogonal factors where we know that they're all going to be orthogonal independent. In this case, actually, when we think about finding patterns in data, often the most interpretable, the most interesting patterns are going to be not orthogonal. In other words, again, we're going to see examples of this in the data, but you can imagine that sometimes these patterns representing interesting effects on the data are going to be very, very well correlated. Okay? And we'll look at examples of that. And we'd like actually to model each of those different components separately. So that's why we want to allow non-orthogonal factors. Interpretability. We'd like some way to understand what the patterns we're actually seeing in the data represent, right? We'd like some way to understand them in terms of the data that we're looking at. Computational tractability, obviously because we're dealing with genomics data, these models, these methods have to be really, really fast. We'd also like to get the dimension from the data. So in other words, the number of factors that we have, this K term, should be inferred from the data. And I'd say this is incredibly difficult. Right now, really the only way to do this, because we don't have statistical tests for these types of models, or we could use model selection, we could use BIC or AIC or something like that. But in general, the way people do it is they run these models for K equals 1 to 10, for example, and sort of choose the one that essentially does the best thing. I've had a lot of trouble using the AIC and BIC types of criteria to diagnose these particular model dimension issues, but maybe other people have not, so you can try that. Yeah, so even when we have these behaviors, we still have to consider the invariances when we think about the downstream interpretation. Okay. I should say I'm a Bayesian, and this is probably fairly unique to what you guys are listening to this summer. Maybe I'm wrong, but I think that's probably true. I think that there is when you think about sort of Bayesian versus classical statistics, I think there's a huge trade-off, right, between sort of the expressiveness of the model and the computational tractability, okay? And I'm interested in building models that are extra expressive, specifically because the applications that I'm really interested in right now, including genomics, neuroscience, medical data, they're not actually big data, right? Big data are when we can keep collecting samples ad infinitum from Twitter or from Facebook or from something else. Google searches where n is really, really, really large. But these are not big data. We call them high-dimensional data because the p, the number of features in our data, is much, much bigger than n, the number of samples that we have. So you think about any genomic study that you know, and in general, there's going to be 20,000 genes, maybe 4 million SNPs. But if you have more than 1,000 individuals, you're really lucky. Okay? So it's a very, very specific application that we're thinking about, in particular these high-dimensional data sets. And the models that are more expressive, in my mind, do a much better job at recovering reproducible signals. So much of my work, and this is not really part of what we're talking about today, but much of my work focuses on thinking about how to structure these models so that you can actually recover signal in this incredibly sample-poor environment. Okay? Then the problem, of course, is how do we achieve computational tractability? Because we have these very expressive models with these highly structured priors. And the answer is that there's a lot of ways to do this that my group is exploring right now. So, for example, we use these nested Laplace approximations, variational inference, method of moments. So we have papers on all of those different things posted on arXiv right now. But we find that we can actually make these models work on really large data sets. So don't be scared of using structure and being Bayesian. OK. So now we're going to talk a little bit about latent population structure. You ready? OK. OK, so latent population structure, again, it really got me into this exploratory data analysis topic. I think it's just so interesting. So it allows us to, it provides us with a bunch of information about DNA. So we can understand, for example, the relationships, evolutionary relationships among individuals in a sample. So, before sort of admixture came along, there was a lot of discussion of the coalescent. Coalescent's been around since, 1980. And up until we had sort of this type of representation, the coalescent was sort of how we describe the relationship among individuals. But now we can actually cluster and consider the relationships of individuals by using these types of latent population structure models. It's much easier, and it does the job for a lot of situations in terms of thinking about how individuals are related to each other. These types of models, the results from these types of models allow you to correct for relatedness in association mapping. So the reason we need to do this is because if you think about, for example, a phenotype that is very different across two populations. So prostate cancer is one that comes up a lot. So African Americans and European Americans have very different rates of prostate cancer. So when you do a naive association mapping with prostate cancer in a sample that includes both African Americans and European Americans, you find that any SNPs that are different in frequency among those two populations are going to come up as hits. Okay? So we need to control for that somehow. And the way that we do that is by using these latent factors. Okay? Another thing we can do is understand population ancestry, history, and ancestry. So we can look at these individuals, for example, and start seeing where the relatedness is from. So one nice example of this is actually from David Reich's group, where they looked very carefully at individuals from continental India and basically describe a really nice method using models that are very similar to this, and actually also using coalescent type models, to describe the different migratory patterns of individuals in India, and also describe the admixture, ancestral admixture between ancestral northern and southern Indian populations. It was very nicely done. And it was using, again, methods that generally look like this as opposed to more rigorous types of models. OK, so the first idea that we can talk about, the first model that we can talk about is an admixture model. So admixture models, and the one that we're going to be thinking about very specifically is one called Structure by Pritchard, Stevens, and Donnelly 2000. They've completely transformed how we think about population structure, population genetics. Structure, as of this morning, has over 17,000 citations, which is completely ridiculous, right? That's a lot of citations. So admixture models are used to do these things that I just said. They're used to explore relationship between individuals, characterize allele frequencies for ancestral populations. So actually, I haven't said this. So actually, one thing that you can get out of these admixture models is for every single population, for each of your K populations that you have in the model, you can pull out the allele frequencies of each of the different alleles for each genotype from the parameters estimated in these models. So for example, if you have the situation like continental India, where no person in India, no person sampled, is either purely northern or purely southern ancestral Indian, you can actually deconvolve those two populations and figure out what the underlying population allele frequencies are in each of the ancestral populations without having a person who's uniquely one or the other. Finally, they allow you to control for latent population structure and association mapping, although I have to say that this isn't really done anymore. We use other methods for that now, principal components analysis mainly. Okay, so the first idea is just this admixture model. So let's just write it out. So let's say we have n individuals, we have L SNPs, I'm kind of changing my notation a little bit, and K ancestral populations. We're going to assume diploid genomes, okay? So there's always 2 copies of any SNP, or 0, 1, or 2 copies. And then basically, this is what the generative model looks like. Alpha is going to be our hyperparameter that describes sort of the spread of population ancestry. So in other words, well, let's go through it slowly. So theta is a Dirichlet. Okay, so theta I is going to be a point on a simplex. It's going to be a k-dimensional vector that describes the ancestral proportions of that individual I with respect to the k populations. Okay, so when I plot the theta term, that's what these destruct plots look like. These sort of rainbow plots that I was showing you. That's plotting theta. So these are individual specific population proportions. Beta over here is going to be a matrix where we consider for each of our k populations. So it's going to be a k by L dimensional matrix. It's going to be the proportion of each of the minor alleles in that population for all of the alleles. Okay. So it's a big matrix. Okay, and then let's see, Z here is going to be drawn from a multinomial. So now the idea is for every single one of my loci, every single SNP in the genome, I'm going to draw 2 Zs, right? And those are going to represent the population that this particular SNP is going to be drawn from. In fact, each version of the SNP. And then we're going to draw 2 X's from a multinomial again with parameter beta that tells me exactly what the probability of each of the 2 different alleles are for the population represented by Z. Okay? Questions? Can I repeat that? Yeah, go ahead.
That's from the zip.
Yeah, I can do from the zip. Yeah, go ahead.
So,
I'll repeat it though.
No, that seems to it's interesting, but then I guess it doesn't really take into account that kind of the genome is transmitted down in chunks. And
Yes.
the nearby pieces of DNA are probably related to each other.
Yeah, it's very embarrassing to give this talk in front of Shriram because he's like done all of this work. So yes, it's absolutely true that it's transmitted in chunks. And right now, yeah, one of the assumptions that we're going to talk about making is that there's going to be independence across the loci. But you're absolutely right. So there are a bunch of different ways to think about how to add those relationships in here, whether it's specifically by assuming chunks, by putting a Markov chain on Z, right? You can do a lot of different things. Absolutely. Okay, so let's go back to what the model means. Yeah? Okay, so going back to Z. So now we have our individual specific proportions, right? So that's the proportion of ancestry. So for a particular individual, I might be 20% European and 80% African, okay? So I'm going to draw Z from that 20-80 split, right? I'm going to flip a biased coin and maybe get African, right, for a particular locus. And so then I'm going to draw X, the genotype at that locus, at a single, one of the 2 copies of that locus, from beta for African. So I'm going to pull the allele frequencies for African at that particular SNP. And again, I'm just going to sort of flip a biased coin based on beta to be able to tell me what allele that is. Does that make sense?
That basically tells you to choose one of 2 populations.
Well, it could be one of K populations. I'm just saying 2 for ease. Yeah. It's going to be one of K populations, exactly. So thinking about it in terms of the parameters that you have after you fit these models, for every single individual, right, for every single locus we have what I sort of think about it as a painting, right? We have some idea of the ancestry of every single SNP in that genome, right? So you can actually sort of, again, paint the chromosome with the inferred Z values to figure out sort of where in the chromosome they are. They have specific types of ancestry. Yeah. Reasonable? Okay, good. Any other questions? All right. Okay, yeah, so I said this already, but I'll just reiterate. Okay, so again, theta is going to be the proportion of individual I's genome that is assigned to each of the different K populations. So I think about that as the population, individual specific population ancestry. And then Z's are going to be the ancestral population of the assignment of just the allele J and SNP L and individual I. Okay, so again, it's going to be a roll of a die where a bias die where each of the sides are one of the K populations. And then beta, again, is going to represent the minor allele frequency of each of the L SNPs in some population K. So it's going to be an L by K matrix. And each of the each of the L rows is going to be on a simplex. Again, non-negative and summing to 1. Any questions? Okay, so admixture models have been used in a lot of different places for a lot of different things. So the original paper, this is Pritchard Stevens Donnelly, 2000. They actually didn't use SNPs, they used microsatellites. So they had something like 72 microsatellites for this set of birds. Each point there is a single bird. And they actually ran this. Some of these microsatellites, I should say, had up to 18 different possibilities. So it wasn't just a 0, 1, or 2. But there's actually 18 different possibilities for the microsatellites. And they plotted the theta variables, the individual specific ancestry proportions on a simplex. So here's 3 of the sort of ancestral populations of birds, right? And you can see that there's a lot of individuals in the corners. In other words, these populations are mostly not admixed. But you can also see that there's going to be a bunch of admixed individuals where they definitely do not have pure population ancestry in only one of the populations. Let's go to this one. So in Rosenberg et al. From 2002, this is an analysis of the HapMap3 data. And this is just one of those destruct plots that you've been seeing a lot of. But the idea is that every single one of these columns is going to be a single individual. And you can see that when, in this case, k equals 6, there's very nice description of each of the populations. And they go through a discussion about how you choose k appropriately, but they sort of come up with 6 for some pretty good reasons. I should say that Pritchard, Stevens, and Donnelly in their paper, if you read it, which I hope you have for your journal club, but if you read it, sort of stop reading it when they start talking about how to choose k because it wasn't really right. But they also have some discussion about how to choose k appropriately as well. So I published this paper with Matthew a couple years ago. And we actually applied these models to the European PopRes data. And again, there's about 1,000 individuals here. And if you sort of squint, you can see that there's kind of a map of Europe showing up there. And I'll make that explicit later. But the reason I think this is interesting is because before this, I hadn't really seen too many applications of these admixture models to populations that were not discrete. Okay, so in general, the assumption is that you're going to have k latent populations, discrete populations. But here is certainly a situation where there's continuous population structure, okay, where there's no discrete populations. And it's possible to do this, although I have to say we played with this a lot in order to get that to work. Okay. So admixture models, like I said, make a ton of assumptions that you should be very clear about when you're analyzing these data, some of which have already been discussed. So the assumption is generally that ancestral populations are discrete, although, like I said, there are ways to do it when you have continuous population structure. The assumption is that the SNPs are independent and identically distributed. This is obviously wrong, and models have been dealt with to change that and allow there to be linkage disequilibrium. Individuals, they make the assumption that individuals are not close relatives. So this is actually fairly important. If they are close relatives, then there's a lot of weirdness in terms of the variables z and the variables beta. locus-specific allele frequencies are all equally likely. So again, the idea is that if you think about the distribution of minor allele frequencies in each of the populations, the idea is that right now there's effectively a uniform distribution on that. You can obviously change that by changing this gamma variable here, but in general, the assumption is that they're going to be uniform. Whereas in practice, in reality, we know that there's, of course, going to be a fairly big bias towards low minor allele frequency SNPs. It also assumes Hardy-Weinberg equilibrium. In fact, it sort of depends on Hardy-Weinberg equilibrium in order to estimate these parameters appropriately. So if there's not Hardy-Weinberg, then there's going to be huge problems in terms of estimating the parameters. So then the question to ask yourself is, when do I use this versus sort of a more sophisticated method for pulling out structure, pulling out latent population structure in the genetic data? So when are they useful and when are they going to be detrimental? Okay, so these are, again, important things to think about. Yeah?
So in terms of allele frequencies,
yeah.
You could sort of, I mean, there's no reason why you couldn't take 1,000 genomes data, estimate allele frequencies, and use those to sort of guide your hyperparameters, but how sensitive is it? You know, I mean, you're saying right now that this is an assumption we're making, but if you could do that or did do that, does it really change it, significantly change your results?
Yeah, that's a great question. So in general, we've found that violating that particular assumption, in other words, the minor allele frequencies are completely skewed, does not affect the data that much. You know, as a side note, the question is, how do you actually evaluate the cost in terms of your results of the violated assumptions, right? So again, I think there's a lot of really nice model-based methods to be able to do that. So the one that I love, because I just wrote a paper about it, is called posterior predictive checks. And so we looked at that very explicitly, making sure that the biases in the minor allele frequencies across 4 different studies were not influenced by this uniform assumption, and in general they weren't. So, but you're right that these things are all important to check, and there are ways to do that. So yeah, so we find it's fairly robust to minor allele frequency. There are other things that it's not robust to. So actually, turns out that choice of alpha actually often has a very big influence on the results that you get, and theta in particular. Okay. Yeah. Okay. So the next idea is principal components analysis. In 2006, Alkes Price and others wrote this kind of mind-blowing paper where they basically took a whole bunch of genomic data, applied PCA to it after standardizing it, and it worked brilliantly. It worked brilliantly in the sense that they really came up with some beautiful understanding of the latent structure for the individuals in the study, and it also had the nice properties that goes along with PCA. In other words, it was very, very fast. You could use singular value decomposition to get it. It was interpretable in the sense that we have this ability to evaluate sort of the p-values of each of the principal components that we identify. They're orthogonal. So there's a bunch of nice properties that go along with this well-studied method that we can use and exploit then in terms of understanding the structure underlying the data. So we can use PCA to do all the things that we do with the admixture models. This is actually not entirely true. It's much, much harder to characterize the allele frequencies, and we can talk about why. But certainly it can be used to control for latent population structure and association mapping using something like Eigenstrat, for example. Okay, so the idea behind PCA is super straightforward. So again, the idea is that if you have a genotype matrix, if you standardize, which basically means for every SNP in your genome, you subtract the mean of that SNP and you divide by the standard deviation. Okay, so it looks like approximately a standard normal. Then you can do singular value decomposition, and you can pull out the principal components from that. And in fact, Eigenstrat, you can do this in a greedy way, and Eigenstrat does that, and it's incredibly fast. It runs, in about 20 minutes on sort of a 1,000 individual by 200,000 SNP matrix that I have. It's super fast, and that was on a computer from like 10 years ago. So conceptually, again, I'm trying to put everything in these probabilistic frameworks. Probabilistic PCA does have a model-based interpretation. So the way to think about this is if you have your genotype matrix, then each element of that matrix ij is going to be distributed just like before as the inner product of each of our k, of the ith and jth loading and factor. And then we have the residual variance term. And the difference between this formulation and the formulation that I showed before is that the residual variance terms are all identical. Okay? So the main difference between factor analysis and probabilistic PCA, again, is that for factor analysis, the residual terms are going to be specific to each j, whereas for probabilistic PCA, there's going to be a single variable. Okay? And that turns out to be incredibly, incredibly important when you interpret these with respect to whether it's modeling variance or covariance structure, which is a subtlety, but there it is. Okay, so so again, the thing about this is that there's going to be a single variance parameter. The k columns of lambda, of matrix lambda, are going to be orthogonal, and the k rows of f, in this case the factors, are going to be orthonormal. Okay? Questions? So the reason this works, again, is just because in this formulation, we can go back to papers by like Sam Roeis and Bishop and Tipping. The reason that this works as a probabilistic interpretation of PCA is because the maximum likelihood solution to this happens to be the singular value decomposition. Okay? Okay, so PCA has been used for quite a number of things, and it works beautifully as well. So in the original Price et al. Paper, they showed how they could cluster European samples. So again, this is assuming that there's going to be a continuous population structure, but they showed in that sense that there's beautiful clustering of discrete populations. Discrete populations. There's this absolutely incredible paper by John Novembre and Matthew Stevens from 2008, where again they took this PopRes data set, which is about 1,000 individuals from across Europe. They ran PCA on it, and they basically pulled out what looks like a map of Europe, okay? Which is absolutely phenomenal. So in contrast to the discrete population structure that we saw for admixtures, this does a very, very nice job in thinking about the sort of population gradients across continuous populations, okay? Gil McVean wrote this really nice paper too, thinking about the interpretation of the principal components for discrete populations. This actually is describing the number of independent SNPs, but I think one of the main take-home points from this paper was describing the relationship between the points, the principal components, and FST, which is basically the distance between each of the populations. So he has a really, really nice interpretation of that, and I highly recommend reading that if you're interested. Okay, so just like all of the other methods, PCA makes a number of important assumptions that are absolutely essential to think about when you apply them to data. So it makes the assumption that population distributions are orthogonal, which is absolutely not true. It makes the assumptions that populations are continuous, which may or may not be true, depending on your data. It makes the assumption that genotypes are standardized. So this actually has fairly big implications. So for the other models I told you about, admixture specifically, but I'll tell you about another model next, the genotypes are not standardized. We use 0, 1, and 2. Here we standardize them. But if you think about what that means for a particular SNP, right? If you have a very low minor or low frequency, if you have very few 2s, then what's going to happen is that when you subtract the mean and divide by the standard deviation, you're going to have exactly 3 numbers. Most people are going to be something slightly below 0. A few people are going to be something way above 0, and a few people are going to be something really, 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 really large. Okay? And if you think about the effect of that on the variance, it actually changes, it inflates the proportion of variance explained that is due to these low minor or low frequency SNPs relative to the genotypes that are not standardized. Okay? So it actually, there are slight differences there. Also, there's no interpretation of the parameters in terms of proportions. So these are all going to be continuous variables. They're not only positive, so you can't just normalize them. So it's pretty important to think about if you actually want allele frequencies for specific populations, that this is probably not the right thing for you to use. Okay, so the third idea is sparse factor analysis. So the idea there is that we're just going to use a factor analysis model that I've been telling you about, and we're going to put a sparsity-inducing prior on lambda. Okay? So sparse factor analysis is a flexible alternative. It makes different assumptions and allows you slightly different interpretations. factor analysis is generally used for exploratory data analysis, like all of these things. Oh, I'm not sure why it says price at all. I think probably all of them do, but anyway. Okay, so here is the representation that I've been telling you about, and we're just going to talk about the priors that we used here. So this is Y, again, our genotype matrix. And we're going to have, again, the individuals on the rows, and the columns are going to be the SNPs. We're going to decompose this into lambda and X plus our isotropic noise, where, again, the residual noise is going to be parameterized for each of the individuals in this case, actually. And just like before, we're going to have the inner product of the ith and the jth components there as the mean. And there's going to be sparsity in lambda. And the way we induce sparsity in lambda, I should say really quickly, we're going to put a prior on X, which is just going to be a normal 0, 1 vector, independent. Lambda is going to be drawn from a zero-centered Gaussian distribution, where every single component, every single ik component is going to have its own residual variance term. Okay, so it sounds like we're way, way, way over parameterizing this model, but actually, if we put a nice conjugate inverse gamma prior on that sigma squared variable, this is something called an automatic relevance determination prior. And it ends up allowing you, with this beautiful conjugacy that we have, ends up allowing you to effectively have lasso or L1 type penalty on the lambda variables for computationally very, very cheap. So we can use variational methods to implement this, and it's super fast. Actually, basically as fast as the principal components analysis that I told you about.
I'm sorry?
Yeah.
What was that? The lambda ik.
Lambda ik. Yeah?
No, the first one. That we keep.
Oh, transpose. Yeah, yeah. All right. Okay, so as with the others, sparse factor analysis is going to make a different set of assumptions. So the idea is that the latent components are going to be non-orthogonal. Okay, so they might be well correlated. And I think actually this is probably a good thing. We have a Gaussian residual function, which obviously plays out in the sense that we're dealing with 0, 1, and 2 variables. And so it could be seen as a good or a bad thing, I think. We can include an intercept term, which allows you to sort of think about the sort of genetic center, if you will, of your sample. The components are not going to be interpretable in terms of proportions, just like PCA, because they're going to be continuous variables, not only positive. The sparsity is only going to be in the factor loadings, not in the factors. For better or for worse, we're going to make the assumption that the structure that we see affects all of the SNPs, not just a subset of them. And then the many invariances are going to be made explicit. So in other words, we talked about rotational invariance, scale invariance, and label switching. So those are all going to be very explicit in here. OK, so let's, in the 3 minutes that we have left, talk about, I'm done in 3 minutes, right? I can keep going.
Actually, this is great. Why don't you just do this and then continue?
Yeah, that totally works. OK, so OK, yeah, let me finish this part of it because this is the punchline. OK, so let's take these 3 different methods. I realize I don't actually have latent Dirichlet allocated or admixture model in here, but I can tell you what it looks like. So let's apply this to the HapMap. Let's apply these methods to the HapMap2 genotype data. Okay, so these are going to involve 210 individuals from 3 distinct ancestral backgrounds. Okay, colored and coded like that. The idea is that when we use PCA, so remember that we standardize. So we're pretending that the first principal component that we come up with is actually just going to be the mean, right? So it ends up sort of effectively being a constant factor. Then the first PC looks something like this, where you see a really clear separation of the 3 populations, right? So basically here we're discriminating African from European and East Asian. And here it's slightly different. This PC appears to separate European from East Asian. And African's already been separated in some sense by the first PC, so this is what's left. So think of it as sort of a greedy thing. And you can imagine if you plotted PC1 against PC2, you would see 3 very distinct clusters, right? 3 very distinct clusters, and you absolutely do. So sparse factor analysis does something slightly different. Since I have a whole bunch of zeros in my loadings matrix, I'm going to get a whole bunch of zeros in my individuals. So when I plot this now, I see that the first principal component pulls out East Asian, and then the second one pulls out African, and the third one pulls out European populations. Okay, so let's talk about this for just a second. In this sense, if you actually look at the correlation of the factors corresponding to these loadings, they are very, very highly correlated. Do you guys know why? What do they represent? What are the factors corresponding to these loadings represent? Am I allowed to be interactive? They represent the allele frequencies, right, for each of these different populations. So the factor corresponding to this loading is going to represent, for example, the allele frequencies for the Europeans. Okay? This one's going to represent the allele frequencies for Africans, and this is allele frequencies for East Asians. They're going to be very well correlated. Why is that? Because our allele frequencies are really well correlated across all these three populations, right? That's really the entire thing. So in principal components, you take out all that shared structure here, right? Anything that's shared, you take out there. But then here, you're only picking up the SNPs that are different, effectively most different, are able to discriminate the African and the East Asian populations. And here, you only picking up SNPs in the factors that discriminate European and Asian. Okay, so they can be entirely orthogonal, right? Because they're not going to be the same. This is entirely non-orthogonal because each of these population frequencies are going to be nearly identical, except for a few SNPs. Okay, does that make sense? Right. People are getting tired. Okay, last thing. Okay, so let's apply this to a continuous population data set then, right? So here again is going to be what the actual map of the individuals is kind of washed out in terms of the color, but map of the individuals from this PopRes data set. There's over 1,000 individuals. We're actually running this on 200,000, like an LD pruned set of SNPs. And you can see, okay, so first we did admixture. Wait, is that right? Yeah, so first we ran admixture, and again, we had to sort of monkey around with how to get this to work, but we ran admixture with 4 different components, and then plotted the geometric mean of those components for each of the individuals. And you can see, actually, you end up recapitulating this map very, very nicely, right? There's going to be much more pushing of the genotypes toward the edges, but I believe that's because there's this binomial distribution describing each of the SNPs sort of in the residual. And that ends up having this sort of regularization effect where things get pushed toward the edges. Here we have the PCA loadings where, again, you can see the structure of Europe come out really nicely. Where, for example, this is Sardinia, I believe. These are Crete. Whoever has explored this data more than I can tell me. Anyway, it's just absolutely stunning the way that falls out. The catch about this, of course, is that if you have double the number of Spaniards in here, then the entire picture warps. Okay, the entire picture warps in the sense that all of a sudden Spain becomes much bigger. So it's very, very dependent on the number of samples in terms of the shape, the number of samples that are represented in each of the different areas. Okay. Here's a sparse factor analysis, and what you can see from this is that, first of all, it looks very similar to PCA, but you can see that there's this X going through it, and that's going to be where the sparsity is. So because we're completely invariant to rotation, every single time I ran this, it was another rotation. And you can see how I rotated it to look like Europe based on where the sort of 0 lines are. Okay? But that said, but despite, there's again slightly different regularization because of the slightly different assumptions, but you can see the exact same set of points are going to be outliers on all the different plots, which is pretty cool. So you can recover this population structure very nicely in continuous and in discrete populations from all of these different approaches. I think that's all I want to say. Yep, I think that's it for now. Thanks.